Good afternoon. Welcome to Medical Grand Rounds. Thank you for being here. Today we're going to do an update on COVID-19, epidemiology, virology, prospects for drugs and vaccines, and then a brief session on UCSF's experience in the Navajo Nation. I'm Bob Wachter. I'm chair of the UCSF Department of Medicine. I'm welcoming a warm welcome to our colleagues in the Department of Medicine, our friends at UCSF, and at our partner sites in the UCSF Health Network. I see we already have about 600 people watching us, and we will post this Grand Rounds as we have the others in this series on YouTube later tonight. The 6 previous Grand Rounds have garnered about 200,000 views, so really quite wonderful. A few quick ground rules are shown here on the screen. You will be on mute. If you have questions, please submit them through the Q&A button. My colleague Quinny Cheng is screening them, and we will try to get to as many of them as we can in the discussion sessions after the talks. None of the speakers have any relevant conflicts of interest. In today's Grand Rounds, we'll begin with a discussion from my colleague Shamasunder, who's one of 20 UCSF clinicians who have been providing healthcare in the Navajo Nation, one of the hardest hit areas in the United States. Last week, I gave George Rutherford the week off, and we saw comments on YouTube saying, "Bring back George." So he will be back today to give us an update on the pandemic, particularly as we enter this next phase of gingerly beginning to open up certain businesses and other activities here in the Bay Area and around the country. That'll be followed by a Q&A. Then we'll spend the last hour, beginning at 12:30, going to about 1:30, focusing on the virus, having some of our world-class experts unlock the mysteries of the virus and hopefully their vulnerabilities, which will ultimately be our way out of this. And I will introduce those 4 experts when we come to that session, again at about 12:30. So the whole session will last about 90 minutes, as is our tradition. And let's go ahead and get started. If we can bring Sri on. So Sri Shamasunder is associate professor here in the Division of Hospital Medicine, and he's director of the HEAL initiative. And I'm sure he'll spend a minute or two telling us about what HEAL is. One of the remarkable things that happened here at UCSF was about 3 or so weeks ago, we all recognized that we were on the lucky end of things and not getting hammered. And immediately as we began recognizing that we weren't going to see the surge that I was seeing, hundreds of UCSF clinicians volunteered to help others. And a group of them went to New York. You heard from some of them a week or 2 ago. And a group went, I guess, a little over a week ago to help out at the Navajo Nation. Sri is leading that group. So we look forward to hearing his comments. Sri, welcome, and thanks for being here.
Thanks for having me, Bob.
So let's start out. Just tell us a little bit about HEAL, because you had an experience and a relationship with the Navajo Nation before all of this.
Yeah, so HEAL stands for Health Equity Action Leadership. It's a 2-year global health fellowship program. Its goal is to train and transform frontline health professionals to make serving underserved populations a lifelong choice. So really getting excellent at taking care of underserved communities. And so we've started HEAL in 2015. And over the last 5 years, we've had 150 frontline health professionals. Half of them are US doctors that are post-residency at UCSF, and the other half are either Navajo or from countries such as Haiti, Mexico, Malawi. So it's this really incredible cohort, and we've been working in Navajo Nation since 2015.
And thank you. What made you think that the Navajo Nation might get hard hit from your experience with them before all this started?
Yeah, I think, one of the things of shelter in place that we've done so well in San Francisco, in some ways in a place like Navajo Nation, it's a flat tax. It's a regressive tax for very vulnerable patients. And it's part of a pattern that you're seeing across the country. You know, the inequities that we have in our health system is where it's laying bare. COVID-19 is laying bare a lot of those inequities. And so, for example, in Chicago, Black folks only make up 30% of the population, but are 56% of the deaths. And in Navajo Nation, or Native Americans in New Mexico make up 11% of the population, but are 33% of the COVID cases. And so when there's this focus of sheltering in place, a lot of Navajo Nation, a third of the population doesn't have electricity, a third doesn't have water. There's 13 grocery stores on Navajo Nation. And you have this expansive definition of family where multiple generations live together. And so, for example, I was on service last week in Chinle, Arizona, and there was 4 generations of a family that were all hospitalized at the same time, a great-grandmother, grandmother, mother and daughter. And so, every time we're taking care of patients, it's really, I took care of 2 sisters a couple of days ago that were both hospitalized. And so you're seeing both the expansive definition of family and an infrastructure that has always been kind of less than for the first citizens of the United States.
So tell us a little bit about first of all, tell us a little bit about the group that went out. So who joined you in this extraordinary effort?
Sure. So, because we've been in Navajo Nation and we have fellows that were international, and a lot of them came back because of travel restrictions. And so we were sourcing them to Navajo Nation. And then our partners really led the discussion. And I got so many emails that said, we really need nurses. We absolutely need critical care nurses, ED nurses, and HEAL. We don't have access to a huge supply of nurses. So I went to UCSF leadership, and it's really this incredible solidarity effort that mobilized very quickly, where 70 nurses volunteered in the matter of 24 hours. And, I think I made the request 10 days before we left. And so UCSF leadership, really leaning into this idea that solidarity is a verb. And it's been so meaningful for the Navajo people that in this kind of dark hour that they're not struggling alone. And so it's 14 nurses, 7 doctors, a lot of them critical care trained or ED trained from the nursing side, because that's really the need. And we've heard that we couldn't have come soon enough, because a lot of the facilities and COVID wards that might have been shut down, or services that might have shut down, could stay open because of our nursing group.
So paint a picture of a hospital there and an intensive care unit there. You work where I sit right now in a 600-bed academic medical center with 80 ICU beds and world-class specialists and everything. What's life like there? And maybe with particular focus on the ICU as patients get sick and might need to go to the ICU. What is that like?
Yeah, I think one of the things that I'm learning quickly is how locally driven medical care, all medical care has the local context obviously in mind. And so I'm practicing we're across the volunteers, there's 3 sites that we're in: one Gallup, New Mexico, one Shiprock, and one Chinle, Arizona. And so 2 are on the reservation and 1 is a border town. And so Gallup, New Mexico actually has some of our former chief residents, Jenny and Mia, and it actually is a referral center in Navajo Nation. So it has a 6-bed ICU. There's a total of 220 beds across Navajo Nation.
How big Sri, how big is Navajo Nation? It's sort of physically and people?
Yeah, so it's 27,000 square miles. It's about 350,000 people. It's about the size of New England, or a little bigger than West Virginia. And, there's the facilities. I think Gallup actually, that they're super competent, the Navajo providers and the permanent staff, but they decided quite early that as soon as they intubate patients, they're going to ship them off to Albuquerque and really keep those 6 ICU beds, because as we know, patients stay for a long period of time once they're intubated. And so we really can't have all 6 beds occupied by intubated patients. So despite being incredibly gifted clinicians, they're sending, and the real fear is like, what happens if Albuquerque or Flagstaff or Phoenix get full? And then in Chinle, Arizona, where I was practicing over the weekend, and I have a night shift tonight, I think that the high flow oxygen, they have like 7 or 8 high flow machines. They have about 30 COVID patients that are in the inpatient right now. And there's a real question whether the mortality, when they are sending patients away to other cities, that the mortality is so high that they've had some good outcomes where they've kept people on high flow and they hit that peak and people transition and actually do okay. So they are kind of trying to thread the needle between when is it kind of safe enough to manage them there and not be too late to have an emergent intubation.
And then in terms of other specialists, do you have access to infectious disease experts and pulmonary experts and intensivists?
So most of them, there's usually like one-off experts that are potentially on site, or they have relationships with in Albuquerque with University of New Mexico and other larger academic facilities.
Got it. So you're talking a little bit about sort of the whole families being in the hospital and sick. Any other sort of when you leave there, what will be the one or two most memorable stories for you?
I think what has really hit home is just the resourcefulness and the resilience of the Navajo people. And as we all know, it is a sovereign state, and they've really led, from the get-go in incredible leadership where the federal response has been slow. And so, some of our fellows are actually taking over cafeterias and are producing their own PPE, like sewing their own PPE. And, it's really a local, regional story of success and a national failure at the same time. And so really trying to think about, IHS, for example, is funded 1/3 the rate per capita as the VA or Medicare. And so how come, our first citizens are not really put first? And this was before COVID-19, and really kind of leaning into this conversation of, as we get therapies, as we get vaccinations, how does that become a priority? Because clearly COVID-19 has showed us that our health system is inadequate, especially for our most vulnerable, and that's true on Navajo Nation as well.
Do you have any confidence that things will change as a result of this? I'm guessing that there are many people in the United States who just heard of Navajo Nation when they saw reports that it was the third most hard-struck area in the United States from COVID.
I mean, I think there has, like, the colleagues here are running a marathon, and I think the UCSF team with them is running a sprint alongside them. And, I think it's a moving target where people are getting exhausted, the volume of COVID patients coming in. But, Gallup, which I'm at right now, has a lockdown. People can't come in and out unless they're, health professionals, and they're really trying to do their best to put in the restrictions, which are hard on this population, but it hopefully seeing an end of this surge. But right now we're kind of in the midst of it, and some of the modeling says that this is going to go well into the summer. So it's really a marathon for our Navajo colleagues, and the fact that UCSF is leading with solidarity, I think, is so important to this community.
Well, thank you, and please thank your colleagues for me and for everyone here. We're grateful to you for doing what you're doing. It's really extraordinary. I remember the first time you came and pitched the idea of HEAL to me. I didn't quite get it, and so it's very clear what you had in mind. You're really making an amazing contribution. This is a beautiful reflection of that. So thank you, and stay well, and we'll look forward to seeing you back here in San Francisco.
Thanks, Bob.
All right. Let's move on to George. So George Rutherford is a professor in the Department of Epidemiology and Biostatistics, directs our Prevention and Public Health Group, and has been the person who's teaching us all about epidemiology and the state of the pandemic. And I think particularly interesting phase that we have entered now as we begin to loosen up some of the restrictions, and George will help us understand what's going to happen.
Thanks very much, Bob. It's a pleasure to see everybody. And Sri, thanks very much. It's really, truly inspirational. So worldwide, there are 3.8 million cases that have been reported and 264,000 deaths. The US has fully 1/3 of the cases worldwide since the beginning of the epidemic, now with 1.2 million and 73,000 deaths. Those are 73,000 deaths in the space of about 2.5 months. And just to help put that into context, the number of American deaths in the Vietnam War was 56,000. Can I have the, and down here, I think you can sort of see what the world's curve looks like. And you can see that you get this sort of sense of plateauing out here. Although this is such an amalgam of different curves, it's hard to really say what's going on. The next one, please. This gives you an idea of what's going on in the United States. As I said, the US has currently about 1.3 million cases reported. There's a huge focus up here in New York. New York, New Jersey, Connecticut, and it really extends all the way up and down the eastern seaboard from Boston all the way into suburban Virginia. There are also cases in Michigan, in Detroit, in Cook County, New Orleans, Miami. The large southern cities have been relatively spared: Atlanta, Houston, and Dallas. And then another large focus in Los Angeles, Orange County, Riverside, San Bernardino, and San Diego counties in Southern California. I'm going to really try and spend some time on the US today. I've talked a lot about San Francisco and the Bay Area. Suffice it to say that the numbers of cases continue to go down. There was a large spike on Tuesday in cases in San Francisco, I believe, courtesy of Diane Havler, who reported all the cases from the Mission study at once, and so that led to a big spike, but we understand that that's a bit of an artifact. I'll let Diane talk about her own study. I'm happy to try and answer questions about it, but I've tried to, because it's really very preliminary, and they're continuing to get results in, I wanted to sort of take a step back from that. The next one, please. So this is what the curve looks like in the United States, with this very rapid rise, which was in New York, New Jersey, and Connecticut starting right around the mid-March, and a continued plateau. There's really nothing to suggest that this is dropping off, and in fact, it's continuing at a fairly flat rate. I think looking at the last 2 days is a little bit, those will catch up soon enough. So where we are is we're in a steady state with about somewhere around 25 to 30,000 cases of COVID being reported daily nationwide. The next one, please. And this, I think, it helps you understand this point. Obviously, there's so many cases in New York that drove a lot of the data, but here, courtesy of the New York Times, I won't be bashful telling you where I got this. On the left are cases in New York. The solid red line is a 7-day moving average. And you can see how it's gone up and down. On the right, we have cases from the rest of the country, and you clearly can see the increasing the increasing incidence of cases from the rest of the United States. The next one, please. Now, where are these cases? Sri's talked eloquently about the Navajo Nation, but look at where these cases are. And honest to God, I know a ton about geography, and I've heard of 3 of these cities before. Okay, so it's out in towns: Truesdale, Tennessee; Lincoln, Arkansas; Dakota, Nebraska; Noblesse, Minnesota; Bledsoe, Tennessee. And on where these are where there have been outbreaks. There have been outbreaks at correctional institutions. There have been outbreaks at meat processing plants. You can see the one in Sioux Falls, South Dakota. And for those of you who are on the carnivorous end of the scale, the food scale, you'll know that all of a sudden it's difficult to get meat. There's a large outbreak on the USS Theodore Roosevelt. This was the one where the captain was relieved of command for talking about the outbreak. And so you can see another one in another couple of food processing plants. There's actually a large one now in King County in the southern San Joaquin Valley in California. The next one, please. So who are the people who are getting sick? I think Sri alluded to this, but I thought I would put it out pretty starkly. This isn't from California. So male, female, about the same. There's a surprisingly large number of people under 50 who are getting diagnosed. And remember, the people who are getting diagnosed still are somewhat symptomatic. Although we really have testing on demand now, that's been a relatively recent phenomenon. So older people are obviously disproportionately affected. These vertical lines that you can see across the horizontal bars or the proportion of the population that those groups represent. If you look down to cases by race and ethnicity, you can see that there's a disproportionate number of cases among Latinos, and also somewhat surprisingly among Native Hawaiians and Pacific Islanders. which are probably largely Samoan, I would imagine. And among African Americans, it's right up about right at the point of what that they represent in the population. Looking across at mortality on the right-hand panel, you can obviously see the large mortality in people over 65, slight male predominance. But if you look by race and ethnicity, it really stands out where African Americans have disproportionate mortality and compared to other groups, and maybe slightly among Native Americans and Pacific Islanders, as well as slightly among Asians. Latinos represent only 35% of mortality, but have a very large number of deaths. So I think as we talk about who's getting this, it's important to remember who's getting it and who's dying from it. The next one, please. So the New York Times on Monday published a set of projections that the White House task force was looking at when we had a White House task force. And this is, they published where to find them, so I immediately went and got them. And these are the projections. Remember I said that we were running at about 30,000 cases per day? By June 1st, they're projecting 200,000 cases of COVID-19 cases per day in the United States, per day. And as you can see, we're not coming and going up and coming down. We're going up, and then we're going to level off, then we're going to go up some more. And this is a consequence of behavior. These are behaviors that facilitate transmission, people congregating together, not wearing masks, doing all the things they're not supposed to do. I think this is really sobering. And the next one, if you look at it, the next slide, please, is even worse for deaths. As you can see, I mean, why they publish these projections where the actuals are 10 times higher than the projected is beyond me. But just looking at the solid red line, they're projecting 3,000 deaths per day by June 1st. 3,000 deaths per day. Okay. I remember, well, just remember that there were 56,000 Americans who died in the Vietnam War and 34,000 that died in the Korean War. We will make that we will hit that number in a single month. So this is serious, serious stuff. And this is going to continue, at least by these projections, up through the summer. And this may not be California, and we certainly hope it's not. I think it may not. I think it's pretty clear that it's not going to be Northern California. Southern California is another issue, and there's a lot of transmission going on in Los Angeles and the immediate surrounding areas. I had a slide that I decided not to put in at the last second, but it showed case rates in Orange County relative to when everybody came to Huntington Beach to go to the beach. This is a disease with a 5-day incubation period. You can pretty quickly see what the consequences of big congregate gatherings like that are. The next one, please. And then finally, I just wanted to add end on the examples afforded by influenza A, the influenza A pandemic of 1918, 1919. We're over here on the left-hand curve. This is the first wave. Okay. And while I would like to think it's gone up and it's going to come down and we're going to have a low point in the summer, in fact, we are going up now. So we've hit the peak. We went down a little bit. We've now plateaued well off of baseline, and we're headed for potentially a very large outbreak in the fall, which will make whatever we've seen pale in comparison. And then there's even a third wave in influenza A from 1918, 1919 that happened in the following spring of 1919. So I think we have to be really understand what's happening. The states that have opened up early, this great rush to recreate get the economy going again, will be paid for in blood, quite frankly, and it's not going to be evenly distributed across the population. It's going to be on the backs of the Latino and the African American populations if current trends continue. So I think we have to be, while it's nice to be enthusiastic about reopening and we're all bored with our houses, just remember that there's a price to be paid. And if we're going to be able to do this successfully, which I sincerely hope we will, people have to wear masks, they have to socially distance, and they have to do things in a way to minimize transmission. So that's my last one. I'll stop there, Bob, and happy to answer questions.
Thank you, George. That was sobering. Start with a couple that came in from the audience. Why have the high-profile projections about the death numbers changed so dramatically for the worse in the last week or so?
Because they realized people weren't really sheltering in place anymore, and all the other states let their foot off the brakes. I think that IHME said that exactly, specifically.
So the earlier models assumed a level of human behavior and or policy that was better than what has happened?
Through the end of May, yes.
Okay. In San Francisco, where we've had a remarkably benign course of things, and people have been, by all accounts, paying attention to the public health recommendations and rules. Why do you think the cases have plateaued? They haven't come down very much. As you know, we hear about new cases who must have gotten infected, 10 days ago or 2 weeks ago, and I'm always thinking, like, who are they and what happened? So
Dolores Park.
Dolores Park. So,
got 2 words for you, Dolores Park. No, there's ongoing transmission in San Francisco. We think that the R naught is about 0.9. So we're slightly below. We're kind of on the good side of unity here, of 1.0 for an R naught. These are people who are essential workers who are out exposed in the field. These are people who are, really have to go out and work every day. These are people who are coming in from the outside. These are people in large households, as Sri was saying, large multi-generational households that pass the infection from one person to another serially. So those are where the cases are coming from. Interestingly, there has been news this week about pediatrics in terms of a Kawasaki-like syndrome and a vasculitis associated with COVID. There are some data that have come out recently which give you a glimmer of hope that maybe this is not hypertransmissible among children. It's only a couple of things, and they're sort of really kind of just diffuse. One's an investigation of a school, a couple of kids in a school in France. Another's the population-based study in Iceland, which found 0 of 848 children under the age of 10 who were infected. I think that's, it obviously has to be repeated about 800 times for us to believe it. But if this is not as hypertransmissible as influenza A in schools, in elementary schools specifically, we may dodge much of the bullet of that second wave.
What would the theory be about why it would be less transmissible among children? I can see why they would get less sick, but why less transmissible?
Well, one theory that I've heard is that children don't have ACE2 inhibitor, ACE2 receptors in their lower airways. And so they would tend to get that was one of the reasons for not getting as sick, that they don't have that they're not localizing it in their lungs. People who localize it in their lungs cough up more virus, they're more infectious. That's, this is all teleology trying to back into the right answer. But it's an observation.
So as you look at the moves that the state of California, I know you work with the state, so you probably have to be a little careful, but the moves the state of California has made in terms of loosening up and recognizing the sort of pressures of public health versus the economy and boredom and all that, do you think it's appropriate, or do you think we're moving too fast?
I think it's fine for Modoc County to loosen up, okay? You can't even get to Modoc County from California. This is the one in the far northeastern corner where Susanville is. You have to go through Nevada to get to the county. So that's fine with me. That's okay.
Okay.
It's like an island, right? Everybody else pointed out
that so is Manhattan. It's like an island. So it's not all that protective.
I think we have to be really, really careful about doing this. Obviously, you have to trade it off against the economy. People will get sick if we have a prolonged depression. That has tremendous cost in morbidity and mortality as well. So it's a trade-off. Just understand that we're trading economy for lives.
Yeah. Why are things getting so much better in New York? Is it when you get crunched, you have the fear of God and people really begin taking it seriously, or is it because people now have antibodies and that changes the R naught?
Yeah, well, some of it, I mean, so that's when you start talking about not R naught, but R sub E, the effective reproductive rate, because the population prevalence of immunity comes into that. Yeah, there's some population level immunity in New York, not very much, probably no more than about 10%, I would guess. But that may be enough to sort of blunt it. Plus, the fear of God's been put into everybody, and you're kind of over a hump. Remember that maybe 30% of people who have had the disease and have antibodies don't have neutralizing antibody. So you have to be, and we don't know which ones are which. So you have to be pretty careful about equating seropositivity to immunity.
Yeah. The last question from one of our viewers was all these curves that we're seeing of excess deaths. How do you read those? I mean, do you think they're COVID? Do you think they're non-COVID illnesses where people are staying away from healthcare? What do you learn from them that you didn't learn from just reading raw death rates?
I think it's a mix. I think there are clearly people who are staying away. I think some of it, as we saw in the case of Santa Clara County, were undiagnosed COVID deaths. Some of them may be undiagnosed influenza deaths. That's why we do follow pneumonia deaths around, in the wintertime anyway, to try and pick up undiagnosed influenza. So I suspect it's a mixed bag. I think we're going to learn a lot more about that here over the next few weeks as we start to send in specimens for postmortem diagnoses of COVID.
Okay, George, thank you so much as usual. I'm sure we'll have you back again in the next week or two because I don't want the hate mail that I received last week. And really appreciate all you're doing to educate us in this incredibly complex and interesting field. It's important that we understand it really well. So thanks so much.
You bet. Thank you.
Okay, move on to virology. And if you think about it, that may turn out to be the most important topic of all, understanding how this virus and viruses work and what their vulnerabilities are and how to begin attacking them, whether it's with drugs or therapeutics. And we've got four world-class experts to talk to us. The first is Joel Ernst, who's professor in chief of the Division of Experimental Medicine at UCSF. I'll introduce the others as they come on. So Joel's going to talk to us about what we know about viral immunity and a little bit about vaccines. So Joel, welcome.
Thank you, Bob. So I'll talk about vaccines first, and I think I'll reiterate some of the points that George just made, and then I think that Melanie will probably also link into some of the same topics. Next slide, please. So first, I just want to emphasize why are COVID-19 vaccines needed. I think that's obviously obvious, but why are they needed so badly? I mean, there are several characteristics of this disease that I think really mandate the development of vaccines. So first, it's highly transmissible, as we know. It's by the respiratory route, so it's readily transmitted in people's in crowded places. And obviously, it's an incapacitating and deadly disease. Another reason that vaccines have a particularly special role is that this is a disease that can be transmitted by asymptomatic and pre-symptomatic individuals. In other words, somebody doesn't have to have a rash or be coughing in order to be a hazard to people around them and to infect them. George, I think, touched on this a bit, but I'd like to emphasize that without a vaccine and without broad immunity in the population, COVID-19 could very well become an endemic infection. For reasons that George alluded to, community or so-called herd immunity is probably pretty unlikely. In other words, to get to the fraction of immune people that would be protected sufficient to squash the ongoing transmission is going to incur a lot of mortality. And finally, as we know, and as we're experiencing, the economic and the human impact of COVID-19 is huge. Next slide, please. So just a few things of what we know about immunity to COVID-19 so far. Part of what we know, or what we think we know, is derived from the experience with SARS, which is the most closely related coronavirus to the SARS-CoV-2 that causes COVID-19. But I think that there are several of those themes that can probably be safely extrapolated. And the first is that protective immunity may be transient after infection. In other words, in people that were infected with the first SARS coronavirus, their protective immunity began waning between a year and 2 years post-infection. And so unlike things like measles or smallpox, where infection confers lifelong protective immunity, coronavirus infections may be an exception. The second thing we think we know, and this is partly from emerging information on SARS-CoV-2, but partly also from other analogous infections, and that is that neutralizing antibodies likely contribute to protection. And I'll go into a little bit of detail about what neutralizing antibodies are. But as George mentioned just a few minutes ago, the presence of antibodies does not guarantee protection. And that is because the routine assays don't measure the quantity or quality of the antibodies. And by quality of the antibodies, what I mean is that neutralizing antibodies are thought to be protective. Some antibodies can be neutral, they can be there, they can be detected, but not actually provide any protection against infection. And in fact, then at the extreme end, some antibodies can actually be harmful. And we know this in the most detail for another viral disease, dengue, which is transmitted by mosquitoes, in which people can have antibodies that actually make the infection more severe and the symptoms more critical. And so the way I've stated it here is that the right antibodies are needed. And so these would be the goals of vaccination. In other words, to confer immunity that is actually superior to what is conferred by infection itself. Next slide, please. So in fact, there are multiple approaches to COVID-19 vaccines, and each of these, as I'll detail in a minute, is actually being undertaken right now. So the multiple approaches include an inactivated virus approach. And so in this case, you grow the virus in culture, you treat it with one or another chemical to inactivate it and make it essentially non-infectious, but without destroying its immunogenicity. And so a good example of this is the Salk polio vaccine. Another good example is this is really the starting approach towards the seasonal influenza vaccine production. Another approach is to make an attenuated virus, and that is by either engineering it or through selection processes, modify the virus so that it can grow but really not cause severe disease. The Sabin oral polio vaccine is an example of this. Measles, mumps, rubella vaccines are other examples. And so these really have quite a long track record. There is actually one inactivated virus vaccine undergoing studies in non-human primates at the moment for COVID-19. The other categories are more contemporary and a little bit more modern and based a little bit more on modern biology. And the next category are two families of nucleic acid-based vaccines, in which the first one is a DNA vaccine. So DNA encoding a viral antigen is injected into usually the muscle. That's translated into RNA and or transcribed into RNA and translated into protein by our own cellular machinery. And so the antigen is produced, we develop an immune response and hopefully a protective response. Similarly, we can start with RNA, and you need to protect the RNA in specific ways to keep it from being degraded. But again, after injection of RNA, our own cells produce the protein antigen and induce an immune response. In neither case do we have a current FDA-approved vaccine based on either of these approaches. The next approach is a purified protein, and we have many vaccines made of purified proteins. The classic tetanus, diphtheria vaccines are purified proteins. Likewise, the hepatitis B vaccine. That can come in the form of a soluble protein, generally combined with what we call an adjuvant. Adjuvants are other chemicals that stimulate the immune system to recognize antigens, foreign antigens more efficiently and allow for greater quantity and higher quality immune responses. The little cartoon on the right there shows a so-called virus-like particle in which viral antigens are constructed in such a way that they're presented in a format and conformation that resembles their arrangement on the virus itself. The current human papillomavirus vaccine is an example of that one. And then finally, the last category is a so-called viral vector delivery of antigen, where a SARS-CoV-2 antigen would be delivered by a virus that wouldn't make us sick, but that can either replicate or not, but can be used to deliver the SARS-CoV-2 antigen to the proper compartment in the immune system in order to raise an optimal immune response. Next slide, please. So the goals of vaccine development are, first of all, safety. Second of all, to generate neutralizing antibodies. And the cartoon that's shown here is a particle of coronavirus SARS-CoV-2. And the blue and green objects are meant to be immunoglobulin G, IgG antibody molecules. And so the logic of neutralizing antibodies is that the antibodies block the interaction of the viral protein with host cell receptors and prohibit it from entry. Now we don't know this, but in some cases, T cells also contribute to protection, especially cytolytic T cells that recognize viral infections, virally infected cells and kill those cells and thereby confer additional immune protection. So another goal, especially in a case of SARS-CoV-2 and COVID-19, is you want a vaccine that blocks infection where the virus enters. In other words, that confers so-called mucosal immunity. Since we know that SARS-CoV-2 ordinarily enters through the respiratory tract, we want immunity that operates right there at the portal of entry and not just in the bloodstream. Vaccines need to generate long-lived immunological memory, superior ideally to what infection confers in the case of COVID-19. And then ideally a vaccine or vaccines will induce immune responses that recognize viral targets that can't mutate to escape, like the flu virus does, requires a modification of the flu vaccine every year. Next slide, please. So there is a considerable amount of activity towards COVID-19 vaccine development, and I've listed what was up to date as of, I guess, earlier this week. There are at least 20 groups working to develop DNA or RNA nucleic acid-based vaccines. One of those is particularly far advanced with an RNA vaccine supported by the National Institute of Allergy and Infectious Diseases. There are about 25 groups working on viral vectored vaccines using multiple different viral vectors. Finally, there are numerous groups working on protein-based vaccines, including protein subunits, together with the adjuvant that I mentioned, and a smaller group, a number of groups working on virus-like protein particles. These are a little bit more difficult to produce and a little bit more expensive to produce. There's certainly the success with the HPV vaccine, and this is certainly the HPV vaccine virus-like particle approach has provided unprecedented success in that disease. But in a pandemic, you have to be concerned about the rapidity of which something can be produced and the cost as well. Next slide, please. So what are the stages of vaccine development? What I've just described really is the discovery and development stage. The next stage in human studies addresses the question, is the new vaccine safe? Can you give this to people without causing undue toxicity? And some of those studies can be done relatively quickly in the case of high frequency complications. Others may take larger ones. The next step is, does the vaccine stimulate immune responses in humans? Next, does the vaccine protect humans from disease in a so-called efficacy trial? And finally, does the vaccine protect humans in the real world? And that is termed vaccine effectiveness. Next slide, please. So other essential vaccine considerations, especially in a pandemic, rapid, large-scale production is absolutely essential. We need billions of doses of COVID-19 vaccines. For the same reason, they need to be economical on a global scale. And finally, they need to be stable for populations regardless of their location. In other words, a vaccine that requires refrigeration is going to have limited capability for dissemination. Next slide. So just to address one last question, why does it take so long to develop a new vaccine? Really, the safety studies in humans require a long period of time, or the longest period of time, in order to observe for complications and make sure that a given vaccine is safe. So I think I'll end there and be happy to take questions during the Q&A period.
Great, Joel. Thank you so much. Took a very complicated topic and made it present in a way that I actually can understand. So that's always my test of your presentation. Thank you. We'll bring you back again in a little bit. Next speaker is Melanie Ott, who my introduction says Senior Investigator at Gladstone Institute of Virology, but according to an email I just received is now the Director of the Gladstone Institute of Virology as of 10 minutes ago, so congratulations on that. She also has a secondary appointment in our Department of Medicine, and she's going to talk to us about some of the issues in virology of the SARS-CoV-2 virus. So thank you, Melanie. Congratulations.
Many thanks, Bob. Welcome, everybody. So I'm going to shift topics now and really focus on the virus itself. Next slide. And the questions here are really, how does the virus get into cells and how does it make us sick? We all know that viruses are quintessential pirates. They cannot survive outside a host cell. They need to attach and enter an appropriate target cell, and once in the cell, need to harness and hijack appropriate host factors to set up little viral factories in the cells that reproduce the genome and churn out new virions that can infect the neighboring cells and support the infection. Next slide. What we know about SARS-CoV-2 is that it is a relatively large RNA virus. It is in fact the largest RNA virus. It has a genome of about 30,000 nucleotides, which is roughly 10 times more than the regular RNA viruses that we are used to see. This genome encodes for 29 proteins, which fall roughly into two categories. The blue category here, the ORF1A and B, these are proteins that are so-called non-structural proteins. And if you go back to the viral factory analogies, these are actually the proteins that build the viral factory to produce the virions. The other category of proteins are the yellow proteins, the so-called structural proteins, that actually form the virions that are produced and secreted from the infected cells. And you might recognize the S protein here, that's the spike protein, that makes the contact. That's the protein prominent on the surface of the virus, gives it its unique shape, and makes the contact with the ACE2 receptor, and is a major target of vaccine development, as Joel was alluding to earlier. Next slide. The production of the non-structural proteins is relatively straightforward. They are produced out of two large so-called polyproteins that are basically chopped up into 16 individual non-structural proteins that form these, what I call viral factories. And I have highlighted here 3 of the major enzymes that are currently drug targets that are in these non-structural proteins. The 2 proteases are basically the cutters that cut these polyproteins, and they're absolutely essential for the virus to propagate. They are one drug target. And the second one is the RNA-dependent RNA polymerase that, together with other enzymes that I have highlighted above the polymerase, forms the replication complex and really reproduces the genomes. This is basically the driver of the factories. And this is the target of the remdesivir compound that has recently gained a lot of attention. Now, this is a relatively straightforward process in replication and production of proteins that we know from other RNA viruses. Next slide, please. the coronaviruses have developed or evolved a mechanism that is unique for the production of its structural proteins. And instead of encoding all of these structural proteins in one transcript and producing these polyproteins, they have actually evolved a mechanism to produce them or to express them all with individual subgenomic RNAs. This is a very complex process. It is probably driven by the need because of the length of the genome to have additional control mechanisms here, but it allows the virus to better titer its production levels of the individual structural proteins potentially to its needs. It also has allowed it to incorporate additional open reading frames into its genome that are marked here in gray, that it potentially laterally acquired from other coronaviruses, and that makes it better adapted to certain hosts. Next slide, please. So the 29 proteins that are encoded have really been the focus of much of the attention. And you will hear more about them in Nevan's talk right after me. What we have done is we have integrated them in a larger project in the lab, which we call the Viral Library Project, where we have started already a year ago to produce systematic lentiviral library expression vectors for viral proteins from most human pathogenic viruses. And they are listed here in different categories. And we have very early integrated the 29 proteins of the SARS-CoV-2 virus together with the SARS-CoV-1 and the MERS proteins into that viral library. And what this library allows us to do is to really look based on the phenotype of the cells that express these proteins to make predictions about what these proteins are doing to reprogram the host cells. We're currently using the 3 coronaviral protein sets to do high content imaging with a robotic microscope to test for exactly what the expression of these proteins are doing to these cells. Next slide. I'm showing you just one example out of the vast data sets that we're gaining here, which is a protein called NSP4, one of the non-structural proteins that form the viral factories. And we have focused here on cholesterol production in the cells that express these viral proteins. And the reason why we focused on this is because cholesterol is a signature that is very highly, or it turns out that many factors involved in cholesterol synthesis are hijacked by the virus when it comes into the cell, and Nevan will tell you more about this. But we also know that the heightened cholesterol content is a hallmark of the aged lung, and we think that cholesterol might play a role in explaining why the aged lung is doing worse in developing severe disease than a younger lung. So what we found here is that the NSP4 proteins of all these 3 coronaviruses are well expressed on the left in red in the cytoplasm of cells. When we now do click chemistry-based chemistry on these cells and look by fluorescent microscopy, we can see that these cells that express the NSP4 protein consistently across all the coronaviruses have heightened levels of cholesterol production in here. And so these are the signatures that as compared to the empty vector, which is basically the control one and is lower. So we're looking for signatures like this, where we see changes in phenotypes, and we're looking at many different phenotypes, where we're looking at changes in phenotypes that are common to the coronaviruses or that are actually specific for the SARS-CoV-2 in order to potentially explain part of the pathogenesis of this virus. Next slide, please. The other big question that we're trying to answer, and I think virologists around the world are trying to answer, is the question, what are really the target cells of the virus? We know that the airway, upper and lower airway tract is definitively the target of the virus, or one of the major targets. We know that it also replicates in the gut, in the brain, in the heart potentially, and in other organs, in the kidney. But I think we have focused, if we just focus on the lung, we will still see that there's a lot of questions that are unanswered. So my lab is trying to answer this question by using systems that we call the 3D organoid system. These are basically little mini organs in a dish that reproduce the cellular diversity of the organ that the complex organ that we have in our bodies, and gives us therefore the opportunity to really ask specifically what cell types is the virus infecting, and what is the virus doing to that cell type in terms of its function, gene expression, and phenotype. So next slide. When we just look at these airway organoids that we are generating out of resected lung material, you can see that they have actually the right diversity of epithelial cell types in them. And here we look in blue for the cells and their nuclei, and in green we have used specific markers for these additional cell types. And you can see that basal cells are highly represented in the organoids. Secretory cells like club cells and goblet cells are also very well represented, and we do have ciliated cells in there also. We have them at very low numbers. Next slide, please. Now, when we infect these cells now with viruses, again, in green, you see an infected cell with stained for a viral protein. We can now start to answer the question, which of these cell types are being infected primarily, and what is the virus doing in each cell type to reprogram these cells. Next slide. We're doing this mainly by single cell RNA-seq, where you can see on the left bars that quantify the viral genomes in the individual cell types specifically, and you can see that they are most highly expressed in the basal and in the club cells, while the ciliated and the goblet cells have lower amounts of virus, but they're still getting infected. And we can then tune into individual genes, like on the right side, where we're looking for, in the individual cell types, where we can look at specific correlations between how much virus is in this individual cell type, and how is it, and what is the correlation to gene expression changes of individual genes in the cell types. And we are looking for specific changes that we observe in correlation with the virus and infection titer in the different cell types that might be unique and not necessarily found in all of them. Next slide. So the data that I've shown you, we have obtained with influenza infection, and we are currently doing the same experiments with SARS-CoV-2. Now this obviously requires a higher biosafety level than what we do with influenza, and I'm showing you here the team in the airborne BSL-3 at Anita Seal's BSL-3 at Parnassus. Obviously you need extensive PPE and respirators to make sure that people are safe to work with the virus. And we are currently actually expanding the existing airborne BSL-3 capacity that is existing at Mission Bay and at Parnassus with an additional BSL-3 at Gladstone that will actually help advance and accelerate the Virology Institute on this specific virus here at UCSF. Next slide. And with this, I want to end. I just gave you a little flavor of the virology research that's ongoing. I want to thank very much people in my lab, QBI, Nevan's group, our clinical collaborator, Merdad, who helps us with obtaining the lung samples, bio and computational help from UC Berkeley, Anita's team at the BSL-3, and help with sequencing from the BioHub. Thank you very much. I'm happy to take questions in the discussion section.
Great, thank you, Melanie. That was terrific, fascinating stuff. Last formal speaker is Nevan Krogan, who is a professor in the Department of Cellular and Molecular Pharmacology and director of the UCSF Quantitative Biosciences Institute. And he's going to talk to us about SARS-CoV-2 and trying to figure out what drugs might work. Nevan, thank you for joining us.
Thank you, Bob. It's a pleasure to be back in this very exciting forum. So I'm going to spend the next 10 minutes telling you about some of the ongoing work in the recently formed QCRG, the QBI Coronavirus Research Group. Next slide. This initially started with 22 different laboratories affiliated with QBI at the beginning of March. And since then, it's expanded to over 40 different laboratories, encompassing essentially hundreds of different scientists. And our Zoom calls became too complicated, so we needed to break up the QCRG into a number of different subgroups. Next slide. Focused on different technological areas as well as different biological areas. And for example, we have drug discovery led by Kayvon Shokat and Brian Shoykat. There's a virology subgroup led by Melanie. And then we have focuses on specific biological processes like ubiquitination, John Gross, and translation, Davide Ruggiero. And actually, we're going to be adding several new subgroups this upcoming week. Next slide. And just to say that we're not just collaborating with people here in the Bay Area and at UCSF, I'm going to tell you about a project that was published just a few days ago that involved scientists essentially around the world at Mount Sinai in New York, at the Pasteur Institute, a scientist in Seattle and San Diego, North Carolina, in England, as well as a number of connections with companies here in the Bay Area and beyond. It's been a real pleasure to see how fast science can move when we all collaborate in a really an unprecedented way in this project and other projects around the world. Next slide. And this is the paper that I want to briefly tell you about. It was published, as I said, a few days ago, where we generated a SARS-CoV-2 protein-protein interaction map, looking at the connections between the SARS-CoV-2 proteins and the human proteins. And then we looked at those human proteins and tried to predict drugs and compounds that could be brought to bear to target those human proteins in a drug repurposing effort to see if they have any antiviral activity against COVID-19. Next slide. So one of our focuses here in the QCRG is really to target the host. There's a number of different studies out there and ongoing screens that they're trying to find drugs or compounds to target viral proteins, and that's great. We're taking a slightly different tact here. We're trying to identify the human proteins that the virus needs and then come up with drugs and compounds that would then target those human proteins. And there's advantages there in that approach. One of them is you don't have to worry so much about resistance. You know, we don't mutate as fast to the virus, so that is not going to be as problematic. Another advantage here is that I think if we look at the host, we could ultimately come up with a pan-pathogenic strategy. We and others have found over the years that different viruses target similar human proteins during the course of infection. So the logic would be if we came up with a host-directed therapy for COVID-19, it could be applied to COVID-22, COVID-24, and other viral infections in the future. Obviously, a disadvantage here is if you're targeting human proteins, there could be toxicity. But one of the things we're doing to try to overcome that issue is to initially focus on FDA-approved drugs or compounds in clinical trials on other diseases that have already passed toxicity issues to try to overcome that particular disadvantage. So in the bottom left here, Melanie told you there's 29 genes associated with the virus. There's over 20,000 genes in each one of our cells. That virus cannot live by itself. It needs our cells, our genes, our proteins in order to infect us and to live and to replicate. And our project identified over 300 human proteins through a protein-protein interaction study that we think are highly enriched to be the virus needs in order to infect our cells. And out of those over 300 proteins, 69 of which had at least one drug or compound that is known to target it. Next slide. So Melanie did a much better job than I could ever do describing the genome of SARS-CoV-2. So in total there's 29 genes. And what we did was isolate them individually, clone them out one at a time. And what we do is put an affinity tag or on a hook on each one of these. And if you look on the right-hand side of the slide, we're expressing these in human cells. We started here with HEC293 cells. And then what you're doing is using that hook to pull out the protein out of those cells and then see what human proteins are sticking to each viral protein. And we identify this using mass spectrometry. And then we come up with algorithms that we've developed and applied to that data to derive what we claim are high confidence quantitative SARS-CoV-2 human protein-protein interaction data. Next slide. So we were the first lab to clone out each one of these genes. We started this at actually in January before it became very problematic. And when we initially released this data on bioRxiv in March, I just tweeted out, said, if anybody who wants these plasmids, we'll send them out. And it was amazing the response that we got. And over the last couple of weeks, we've actually sent out these plasmids to 318 different labs in 38 different countries, no strings attached, no MTAs. I said no lawyers are going to be involved. We're just sending these out immediately. And we said, please feel free to distribute. So these plasmids, I just wanted to say, spread around the world a lot faster than the virus did. And we're very happy to be contributing to the research that's been going on with COVID-19 around the world in this way. Next slide. So here's the protein-protein interaction map that we released a few days ago. 332 SARS-CoV human protein-protein interactions, including, as I said before, 69 druggable host factors. In this particular representation, the red diamond are the individual viral proteins. The circles are the human proteins, and if the circle is orange, it's known to be a drug target. And I just want to highlight that we've worked very closely with this company, Zoic Labs. They took our data and they did very interesting visualization of it. It's in a very intuitive and searchable format. And I know a lot of scientists are enjoying looking at the data in this way. So I'd encourage you, if you're interested in this data, to go to our paper and click on that particular link so you can peruse the data for yourself. Next slide. So we're working with fantastic chemical biologists here at UCSF, led by Kevin Shokat and Brian Shoykit. They looked at this map and they identified 66 targets corresponding to 69 different drugs and compounds. About 30 of them are FDA-approved drugs, about a dozen are in clinical trials, and then the rest are being looked at preclinically. So we were able to test about 2/3 of these in virological assays with our collaborators in New York and Paris. Melanie has told you about the great system she's got set up. We're starting to collaborate with her, obviously, now. But over the last few months, we've been working with these collaborators that we had set up over the last few years with QBI. Next slide. These are scientists at the Pasteur Institute in Paris, including Marco Vignuzzi. He was born in Italy, but he was raised in Canada. So he's Canadian in my mind, and therefore a great collaborator. And then also Olivier Schwartz and Christophe Donfrey, they're great to work with. And then at Mount Sinai Hospital, Department of Microbiology, one of the best virologists in the world and one of my best friends, Adolfo Garcia Sastre. So we've been working with him as well, testing these different drugs and compounds. Next slide. And just to go into the details very briefly of the assay, they're different assays, but they're complementary. And it was very reassuring to see similar results with 2 different assays in 2 different labs and in 2 different continents. So we're looking here at Vero6 cells, African green monkey cells. There are now human cells that are looking very promising that we can infect and do these type of screens, but initially they were just using Vero6 cells, growing up the cells, 2 hours before infection, adding a drug, then infecting at 2 different MOIs. It's 4 times higher in Paris compared to New York, letting the assay go for 2 days, and then carrying out a formaldehyde fixation, stopping the experiment, and then analyzing the virus. So the virus, as Melanie told you about, is comprised of protein and RNA. So in New York, they used the microscopy setup to look at the virus using an antibody against NP. And then they did a virological assay, a TCID50, looking to see how infectious the viral particles were left in the assay. In Paris, they didn't look at the protein, they looked at the RNA component of the virus via RT-PCR, and they also did a viral plaque assay. So different assays, but very similar in terms of the readout. Next slide. So as I said, they looked at about 40 in each place, and in total, we tested about 2/3 of our predictions, 47 tested in total. Next slide. And we're very excited to report that we've narrowed down on 2 different classes of drugs. Okay, the first one, a set of drugs targeting protein translation. Right? These are proteins that are required to make other proteins, so the virus needs that machinery in order to make its own proteins. And the 2 compounds that we looked at in great detail that showed very promising antiviral effects. One is zotadifin. This actually targets a protein EIF4A, and we actually found its regulator in our map, EIF4H. It binds to EIF4A, so we predicted this one potentially has an effect. This is actually in a clinical trial right now for multiple myeloma by the company Effector. Some of the co-founders are connected to UCSF, including Davide Ruggiero and Kevin Shokat. They're starting a clinical trial right now on this drug for COVID-19. And then the other one was ternatinib-4. This is a compound synthesized by Jack Taunton at UCSF that targets another translational protein, EIF1A. And it's known that there's a structurally distinct compound called pledepsin that targets that same protein. And this is actually a drug that's approved in Europe for multiple myeloma as well. And this company has started a clinical trial using pledepsin for COVID-19 as well. So we're very excited about the protein translational drugs. And then the other set of drugs that we're focused on are ones that modulate sigma R1 and sigma R2. Now, these are two receptors that have been worked on for decades. They're still considered orphan receptors because they're not completely understood what they're involved with. They're connected to a number of different biological processes. But looking at our map, Brian looked at these and said, oh, there's some known drugs and compounds that are known to bind these receptors, including antihistamines, antipsychotics, antimalarials, the hormone progesterone, the female hormone, the anti-anxiety compound. And if you looked at melphalan, this antipsychotic, we're actually very excited about this particular one, this antipsychotic, because it has very potent antiviral activity, and you can get to very high concentrations in the blood in people. Next slide. So just very briefly showing you the inhibition of the protein abiogenesis factors using ternatin-4 and zotafafin. So just looking in the top left-hand quadrant there of these graphs, in red here, we're looking at infectivity. In black, you're looking at cell viability. So essentially you want the black line to be across the board, having no effects with respect to the drug, and then the red line to drop down. And going from left to right on the x-axis, that's increasing amount of concentration. So these are looking very promising with respect to having a strong antiviral activity. Next slide. And then here is some of the data of our modulators that bind sigma R1 and R2, again, showing very strong antiviral activity. You see hydroxychloroquine is here, which is quite interesting. It does have antiviral activity in our assay, but in data that I'm not going to show you, we actually showed that it binds very tightly to the receptor of the heart, hERG, much more tightly than sigma R1 and R2. And this is a collaboration with Brian Roth. And as you guys probably know, hydroxychloroquine has been discontinued in clinical trials, mostly due to cardiotoxicity. But these other compounds and drugs we have actually don't bind to this receptor to the heart. So we think they're going to have less problems with respect to toxicity. Next slide. All right, what's next? All right, we've shared this information via their publication. We've talked to different government authorities about this, different public health officials. We're continuing to work generating more of these maps. We're starting to do combination therapies here in the lab, looking at combinations of these drugs and compounds, also incorporating in remdesivir as well to see if there's a stronger antiviral effect as well. And we're discussing, in many cases here, expediting these findings into clinical trials. I alluded to some. We're also talking to a number of pharmaceutical companies with respect to these sigma R1, R2 modulators. And then the last point there is, I just wanted to say that it's been really fantastic to see how fast science can move when people collaborate. Next slide. And we've really been breaking down silos, not just in the context of QCRG, but with scientists around the world, across different laboratories, across different institutions, and between academic institutions and pharmaceutical companies. Next slide. And to me, this is the most exciting part of this. The question I have is, why aren't we doing this more often? You know, the problem with science is it's so siloed and it's set up in a way which, in my opinion, really discourages collaboration. Well, everyone sees how fast we can move when we can work together. So the challenge now is to keep this infrastructure in place so that we're in a much better position to tackle COVID-22, COVID-24, other viruses, or just other diseases that we ultimately want to focus on in the future. And that's, to me, one of the most exciting aspects of what I've seen over the last few months. Last slide. And then the longer term is to connect the discovery research world with the clinical world and the immunological world as well. That's some longer term plans. And then this is the last slide. If you want any more information, please go to our website, theqbi.ucsf.edu. Thank you very much.
Nevan, thank you. We often talk about sort of the inspiring stories on the clinical side, but the story of what you and your lab and your colleagues around the world accomplished in really a few months is equally inspiring. So thank you. So we're going to bring on the virology, the rest of the speakers. That is Joel, Melanie, and Don. And let me introduce Don, who I have not. Don Ganem is Emeritus Professor in our Division of Infectious Diseases, and quite relevant to this, also led infectious disease research at Novartis, one of the biggest pharmaceutical companies in the world, for 7 years until 2018. So let me start off with you, Don, and just react to what you've heard and put it in the context of what this would all feel like if you were at one of the big biopharma companies trying to think about drugs or vaccines.
Yeah. So thanks, Bob. It was a lot of fun listening to that. So I'll talk about these things in two separate buckets, if I may, the vaccine bucket and the therapeutics bucket, because they're pretty distinctive. Vaccine production and I think in both cases, we can talk about a good news and a bad news situation. For the vaccine development, the good news is, that this is one of the rare cases where every single one of the modalities that Joel mentioned as being theoretically possible as a vaccine is actually being approached, in an industrial setting. And in fact, even better than that, recognizing the global importance of this, some people like the Gates Foundation and Bill Gates himself has been quite ahead of the curve here in anticipating production problems when you're operating on a global scale. He's already started to worry about whether we're going to have enough vials for vaccine to be dispensed into. When companies make plans to roll out these products, they have to make a manufacturing plan. They have to make a distribution plan. They have to deal with supply chain issues. And it often comes down to vials and rubber gaskets and the size of the vial necessary to accommodate the injection, single-use dose vials versus multiple-use vials. Do you want to prepackage in a syringe or not? These are the things that academicians don't think about, but they really greatly affect the ability to rapidly get the product to the people who need it. And Gates has allocated a couple $100 million to anticipate these issues and to try to make sure that when vaccines become available, those parts of the vaccine supply chain, which is known in the trade as fill and finish, can accommodate the products that are coming their way. Knock on wood. So that's the good news. There are over 90 different companies and entities making these different approaches that Joel outlined. I've never seen that before for any given vaccine. So that's all to the good. The bad news is, and Joel touched on this, the more you know about the coronavirus family, the more difficult you realize the production of a vaccine may be. So I did some rooting around in the last few months now that I'm a retiree with nothing else to do. And when you look back over the coronaviruses that caused the common cold, I was excavating old papers from the 1960s that looked at inoculation trials in which people would be given a common cold virus, get a cold, recover from the virus, have neutralizing antibody demonstrable, and then they were re-challenged with OC43, one of the common cold viruses. And 6 of the 8 people who had antibody responses developed a common cold again. Okay? That is not the kind of thing you want to see running into a vaccine project. So the situation is those common cold viruses are a different phylogenetic clade from the ones in the SARS cohort. In SARS, we have slightly more encouraging news. The neutralizing antibody stays up for a year or 2. But again, we don't really know what the role of neutralizing antibody is in protecting a human being because the SARS epidemic disappeared before we could have clinically meaningful challenge results with the vaccine. So I have to say, in this virus family, the prior track record of producing vaccines has not been good, and there are issues. The other thing is the downside issue that Joel raised about antibody-dependent enhancement. In the coronavirus family, we have no human examples of antibody-dependent enhancement, but there is a famous coronavirus. The coronavirus has produced a lot of veterinary illnesses, and there's a common coronavirus of cats called feline infectious peritonitis, and feline infectious peritonitis is characterized by very strong antibody-dependent enhancement. This is something that happens typically when when the virus targets macrophages and dendritic cells because they have Fc receptors on them. So an urgent question for academicians is, are macrophages or dendritic cells targeted by SARS-CoV-2? And this is something that needs to be addressed. And the best, fastest way to get that answer is going to be by studying autopsy material by immunohistochemistry. We need to know, do alveolar macrophages get infected? Do peripheral macrophages get infected? Do dendritic cells get infected? And we should not be doing this in tissue culture. The history of virology shows very clearly that, that when you study this in tissue culture, you often get the wrong answer. You really want to go right to the primary organ and ask, in the real human infection, are these cells targeted? If they're not, then I think we have a lot less to worry about with antibody-dependent enhancement. But if they are, this is going to be an issue. So that's the bad news with respect to vaccine production, is the prior precedents are not promising. Hopefully they won't apply here. None of that should slow down anything that's being done, but it is something that keeps me up at night a little bit. On the therapeutic side, again, we have a good news, bad news situation. The good news is that there are 2 classes of things that are out there right now being experimented with that look promising. Remdesivir has already shown most of the promise that it's going to show. It is an effective antiviral, but it has a major limitation. It's a prodrug nucleotide, and it's not orally bioavailable. So the fact that you have to use it by intravenous administration means nobody can get this drug until they're sick enough to come into the hospital. That typically happens 6, 7, 8 days into the illness at a time in which new infectious virus production is not high. So we're using this antiviral, this fairly significant antiviral at a time in the infection where viral replication is less prominent in the natural history of infection, and we're more starting to see an immunopathogenic or inflammatory consequence of infection. So we need potent antivirals that can be given orally. No point getting another intravenous compound here. We need orally bioavailable drugs that be given when people are first getting ill, because that's the time in which nasopharyngeal is probably at its peak. I'm not saying there's no replication later on. There almost surely is. That's why remdesivir has the effects that are demonstrable. But the effect on survival was not great. And there was an earlier trial that showed no effect on survival at all. I think this is because we don't want to be using primary antivirals out that late in the natural history of infection. So the good news is that remdesivir works a little bit and tells us that orally bioavailable replication inhibitors could do a lot better. The other class of drugs that are out there we haven't talked about are neutralizing monoclonal antibodies and convalescent plasma. There are 2 companies at least that have highly potent neutralizing antibodies out there, and those are going to make their way into the clinic. The question is, where's the best place to use them? If you look at the history of antibodies in therapy, for most infectious diseases, antibodies work much better for prophylaxis than they do for therapy. If I had to be in charge of these programs, I would be wheeling out those antibodies primarily for prophylaxis of high-risk patients in the nursing home setting and in these congregate settings where you have lots of susceptible people. I'd be looking at them to prophylax workers in a nursing home, the staff in a nursing home, highly exposed ER docs in Brooklyn and Queens, those kind of settings, because I think the likelihood that antibodies would be protective there is fairly high. Although we're talking here about protection against a mucosal infection, not as an intravenous infection. There's very little viremia in this condition. And people have said, well, look, they work therapeutically in Ebola, but Ebola is irrelevant. Ebola is a highly viremic infection. Of course, adding neutralizing antibodies to the circulation of somebody who's highly viremic is going to be beneficial. When you're talking about a respiratory infection, the situation is much more complicated. If you look at the one case where we have an example, which is RSV in children, monoclonal antibodies to RSV are about 50% effective when given as prophylaxis and completely ineffective when given as therapy. So I don't say we shouldn't be attempting them as therapy. We should. They may have some beneficial effect there. But I would, if I had to bet my kids' college fund on this, I would bet on prophylaxis. Of course, my kids have both graduated, so it's a low risk bet for me.
Thank you, Don.
Yeah.
Let me move on a little bit. That's really interesting. We're hearing lots of reports about mutations, and mutations may be making the virus more deadly, more contagious. Then we hear about SARS, quote, dying out, and the 1918 pandemic died out, I guess, at some point. Can somebody sort of tackle the issue of mutations? And is this hyperbolic to hear about this super mutation? And then when a pandemic dies out and it's not from herd immunity, what actually happens to make that happen? Don, do you want to start with that, and then we'll sort of see if others want to chime in?
Sure. If you look at the other coronaviruses, there are lots of examples of mutations that occur that alter tropism or alter pathogenicity. There are many veterinary coronaviruses where that has been known to happen, and causing something that began as an enteric pathogen to wind up as a respiratory pathogen. So the evidence that it's happening now is one recent paper that's gotten a lot of attention. It's too preliminary to say whether that really is indicating what the author speculated it's indicating, but it's a possibility. I don't think it's something that is going to affect any of our therapeutic strategies or any of the immunoprophylaxis, because it's not about an epitope. So I don't think it changes any current research agenda. It's an interesting theoretical question.
And Yeah, go ahead.
how epidemics end. The SARS epidemic was ended by classical, containment. But the flu epidemic, the 1918 flu epidemic, did not end. The 1918 flu virus that caused all of that damage became the prototype H1N1 influenza that circulated for 10 or 15 years thereafter in the face of partial immunity and further mutation. So in that case, it was ended largely by partial prior immunity and probably drift away from more pathogenic consequences. But that virus did not disappear. It became the prototype of seasonal influenza for a couple of decades thereafter.
Great, thank you. Joel, someone asked about vaccines and immunocompromised people and the elderly who have weaker immune systems. They're concerned about whether even an effective vaccine will work in the most susceptible populations.
Well, it's clear that from many other vaccines that many vaccines are less effective in older people than in younger people. I think that's a huge concern here because of the disproportionate mortality, just like it is really mimicking flu. I think one of the, kind of referring to Don's approach to the good news part, I think out of the 90 plus vaccine efforts, I think we're going to have multiple vaccines emerge. And I think some of those vaccines are going to be more appropriate for elderly people. Some of those vaccines are going to be more appropriate for other populations. And so I think that's going to take time to learn, but I think that there should be encouragement. We've learned recently that there are two ways to make the flu vaccine more effective for elderly people. Use a higher dose of the vaccine or to incorporate an adjuvant, which hasn't been included routinely. And so I think we're learning, and I think we're paying more attention to how to make vaccines more effective for elderly people.
Right. Melanie, your discussion of the target organs I found interesting. Tell me, why do we care where it goes or lives, or is it because it can find sequestered spaces and hang out in places that will make it difficult to do treatments, or are there some other reason that it's useful to know kind of what it specifically does in different organs?
Well, I think it's very important to know where it hangs out. I think we know, for example, that the ability to infect the lower respiratory tract might be correlated with the severity of disease in some patients. And so I think it's going to be very important to find out what cells are going to be infected there and how they're contributing to severity of disease and pneumonia. I think what you're alluding to is sort of immune protected spaces that where the virus can hang out and undetectedly replicate. We haven't really looked at these. These are classically sort of in the eye or in the testes. That's where viruses have been found. I think people have not yet looked very closely there to see whether there's continuous replication there. I think what we can say is that the virus can replicate in many cell types. I think it's important, for example, in the gut, it can very effectively infect the enterocytes, but we know that we have tons of RNA in the stool. We also know that the stool is non-infectious. So I think we have to learn what in there is making the virus, non-infectious. Is it the stool, or is it the production of viruses from certain cell types that might be different than in other cell types?
Thank you. And this maybe, I don't know if it's to Nevan or you or really any of you, but this is a question from a political science major who, there's a level of sophistication here that I don't think I have. When we hear about the importance of mucosal immunity and the mucosa as the entry place, and we shouldn't touch our nose and touch our eyes and all that, in my naive sense of the world, I wonder, why isn't there some sort of nasal spray or something mucosal that I can do to prevent this thing from entering me in the first place? The blood seems like a end around to get at what we're trying to do. So maybe Melanie start and others can chime in. And if it's a stupid question, go ahead and just tell me.
No, I think I would just say very briefly that I think this is something that many people are currently looking at, because I think it would be the easiest way if you could take one dose of an inhalant in the morning and you would be protected for the rest of the day or for longer. I think that's definitively something that people companies and academicians are looking into. So it's not a stupid idea at all.
Okay.
I think the blood is really not the route that we should be looking into. I think it is, coming through the respiratory tract. And I think for the secondary infections, I think we have to find out how it's being transported to the gut and to other organs that it eventually infects in the brain. I think a very, very important focus currently of pathogenesis. So these are all important questions, but what Don said is really true. I think the blood is not the place to look for large quantities of the virus. I think it's in the nose, and it's potentially in the saliva.
Interesting. Anybody else? Nevan, you want to add anything to that, or Don?
No, just to add, I mean, that's a question, I guess, related to some of Melanie's work with the organoids. I think it's so important to study this virus in all these different cell types and as a physiologically relevant system as possible. So to molecularly study this virus and all these different organoids is just going to be amazing and fascinating and give us great insight and hopefully point us in novel therapeutic directions.
Okay.
Yeah, Bob, I'll just say about the nasal spray, it's largely a pharmacokinetic issue. It's hard to maintain therapeutically significant levels in the upper nasal passages by nasal sprays alone, because they rapidly get diluted out and swallowed. And just as a prophylactic strategy, it's probably difficult. As a therapeutic strategy in early infection, it might have some value, especially when we're sending people back home to be in home quarantine and there's a risk of spread to the family. There might be a way to deliver things that way, but it's mostly a pharmacokinetic issue. But I think the other thing that comes to mind about the practical value of knowing all about the tropism here, and this relates to something Melanie just said. I just saw two interesting papers about the fact that the GI infection is clinically insignificant and there's no infectivity in the stool, but there's lots of RNA in the stool, and people have been taking advantage of this to monitor sewage, to monitor wastewater. And there's a very interesting case in Barcelona where they looked retrospectively. And since they knew where the sewage was in various parts of town, it is collected from different parts of the municipal water supply. They were able to correctly locate a week or two ahead of an outbreak of respiratory disease just by doing deep sequencing on municipal wastewater, neighborhood by neighborhood, who was likely to have the next outbreak of respiratory infection. That's one isolated example, but it might be a very useful way to be monitoring very big urban populations that, that would allow you to anticipate where you might want to step up testing on respiratory secretions and antibody testing in the sort of like the old cholera epidemic around the pump handle kind of setting.
Yeah, let me ask one, maybe two more questions. We'll go for a few minutes over. It feels like, we talk about the fastest vaccine ever discovered took 5 years, and finding therapeutics that work for HIV took 10 years. Other than the fact that we're going to spend $1 bazillion on it, and the kind of team science that Nevan described is happening, and that Gates is involved, and governments are spending whatever it takes. Can you name one scientific advance in your field that we didn't have, let's say, 5 to 10 years ago, that you think will accelerate either drug or vaccine discovery? Maybe Joel, do you want to start with that?
Sure. I would probably make the point that our understanding of innate immunity and the role of adjuvants has been huge, and there has been a huge amount of attention to developing and characterizing adjuvants in order to induce particularly desirable immune responses for specific infections. So I would say that that's probably one of the advances that may very well make the biggest difference in accelerating vaccine development.
Nevan, do you want to tackle that sort of in your world? What's the big scientific advance that really made a difference here?
Well, I guess for me, I kind of touched on this in my talk, is the silos that have been really broken down. It's, the structural biologists talking to the virologists, talking to the clinicians. I haven't seen that before.
Yeah.
And this is the way we should be doing science in the future. So there's been an unparalleled communication across these different levels, also with academia and pharmaceutical companies. Yeah. So for me, I haven't seen that before, and hopefully we can sustain that type of infrastructure going forward.
That's really comforting. I would have thought you would name some $10 million machine, but do you think it's actually the people talking to each other that's the biggest thing?
It's not the connections between the genes and the proteins, it's between the people. That's the big breakthrough here, in my opinion.
Right. Melanie, any one from your world?
I agree, but I want to bring it back a little bit to the machine. I think I have done research in the early AIDS epidemic, and I have done research here, and I think we have differences how we do research and how we communicate. And I think Nevan is really an excellent example on how the silos have come down and how communication and collaboration has increased. But I think we're also doing science differently, at least from my world, where we're looking more holistically and in a systems-based approach at everything instead of just at one thing after the other. And I think that combined with enormous computational enhancement of computational capacity has really helped us to go much faster today than we did 30 years ago.
Don?
Yeah, I think anyone who's ever worked in industry knows that that kind of interaction is absolutely essential in making a drug. And culture trumps technology in any intellectual enterprise. Culture trumps technology. You have to have a culture of rapid dissemination, among a group of people who are already technologically sophisticated. You just have to break down the barriers between how they communicate with one another, how they understand one another, and how they value what one another does. And
Right. I would end here, but there's one more burning question I have to ask all of you. So take out your crystal ball and tell us when there will be a game-changing either drug or vaccine. Which one's going to come first, and when it will be? And by game changer, I mean it lowers if it's a drug, it lowers the probability of getting infected or getting sick so much that we can go back to something resembling normal life. Or a vaccine, it works at nearly 100% effectiveness, and it's broadly distributed. So which one is going to happen first, and give us a date when you think that is likely to happen. So Joel, do you want to tackle that one first?
Sure, July 14th, but I won't tell you what year.
That's not helpful. And which is it, a vaccine or a drug?
I think drugs going to probably come before vaccines because I think because of the safety requirement for vaccine testing.
All right. And I need a year. 21, 22, 20?
21.
21. Thank you. Melanie?
I agree. I think the drugs are going to be faster. I think they might not be perfect, but I think we will have hopefully more than one candidate that we can combine in the future. And I think that will make it better, as we have seen with HIV, and that will slowly become a game changer. And I would say beginning of 21. Beginning of 21. All right, Nevan.
So I agree with Melanie on the combinatorial aspect of this with respect to drug treatment. And, I think there's going to be a drop here in infections and then a spike in the fall. So I'm going to be more optimistic, considering all these great minds collaborating, working together. I hope there's a more powerful drug regimen by the end of 2020.
All right, terrific. And Don, I'll give you the last word.
Well, I'm going to be the skunk in the
one
garden
who knows how hard
here.
this is coming out of pharma. It's not just a matter of finding the compound, but getting into every.
I'm going to be the skunk in the garden party here. You're talking about a game-changing drug or vaccine. I don't think that repurposing is going to give us a game-changing drug. That's asking for an awful lot. The best we can hope for from repurposing is a partially effective drug that maybe limits mortality somewhat. But I don't. If I had to bet on game-changing result, I would bet that the vaccine happens first. That a partially effective vaccine would be a much bigger game changer. And the prospects for it are better than something emerging from repurposing. Drugs have to be tailored to the purpose they're being built. That's the one thing I learned in the 8 years I was in a company. Hits are easy to make. Micromolar inhibitors, which is what usually comes out of a repurposing effort, are easy to make. But those are rarely game-changing events until you get to the nanomolar potency level. So I would bet that a partially effective vaccine might happen in three years or so, given the magnitude of the effort, and that'll beat a game-changing drug. I'll bet we might get some partially effective drugs out of repurposing.
Vaccine in 3 years. All right, fasten your seatbelts. Thank you all for a really fascinating discussion. I learned a ton. I'm sure people who were listening did as well. And I wish you all good luck and continue to do great work, because we need it. So as I mentioned earlier, this video will be posted on YouTube later today. I'll disseminate the link on Twitter tonight and through more traditional channels later. Thanks to our editorial and production team. You see they're listed. We'll see you back next Thursday. I know we'll have a group of the intensivists who've been working in New York to give us an update on their work. We'll talk some about the I believe we'll talk some about the Mission and Bellina study as well, and then I'll scour the universe to try to figure out what else is interesting and important. There seems to be new stuff every week. And as I've said, we'll continue these grand rounds every Thursday until there's no longer a need to do so. Hopefully that will be sooner rather than later. So until then, thank you for joining us and stay safe.